Hi, everyone. Thank you for joining us. I am Katie Sacro, Director of Family Programs here at the GFPD, and I will be your host for tonight's webinar, Towards Retinal Gene Therapy for ZSD, an update. It is my pleasure to introduce our presenter this evening, Dr. Catherine Argyriou. She will be here sharing insights into gene therapy, including the latest advancements in retinal gene therapy and the path toward clinical trial. Dr. Catherine Argyriou completed her Bachelor of Science degree in genetics with honors at the University of Manitoba, followed by a Master of Science in molecular biology and her PhD in human genetics at McGill University. During her PhD, Dr. Argyriou spearheaded a preclinical retinal gene therapy trial in a mouse model of peroxisome disease, attracting early-stage venture capital investment. As a postdoctoral fellow, Dr. Argyriou championed the further development of retinal gene therapy for PEX1-associated Zellweger Spectrum Disorder, aiming to transition her research from academic proof of concept to treating patients. By working with a local industry partner, she expanded her skill set beyond the laboratory bench to developing a clinically translatable product. In her new position as an assistant professor at the Children's Hospital of Los Angeles, University of Southern California, Dr. Argyriou's goal is to develop targeted gene-based therapies for a personalized approach to treat rare ophthalmic diseases and to study the effects of peroxisome dysfunction on disease pathophysiology. Dr. Argyriou is undoubtedly familiar to many in our audience tonight as a dedicated member of the GFPD Medical and Scientific Advisory Board, which consists of internationally renowned researchers committed to improving the lives of individuals with peroxisomal disorders. Following her presentation, you'll have the opportunity to engage with her directly in a live Q&A session, but please go ahead and submit your questions throughout her presentation. You can do so at any time in the box marked Q&A, which is located beneath the webinar player window, or I'll be monitoring the chat box with Brian Maple, so you can submit there too. And Dr. Argyriou, thank you for being here with us tonight to share with all of us, and I'm going to turn it over to you.
Thank you, Katie, for that lovely introduction. I'm very happy to be here with all of you tonight, coming at you from still Montreal, Canada, through your computer screen, but soon to be coming at you from Los Angeles. So what I will discuss today is probably familiar to a lot of you that have been around the GFPD conferences for a while, but we do have a few small updates that I'm happy to share with you. And as many of you know, project very near and dear to my heart for the last more than several years, many years, has been developing a retinal gene therapy for Zellweger Spectrum Disorder with the goal of improving or preserving vision for people with ZSD. So to give an overview of what to expect today, I'll start by describing what gene therapy is in high-level terms and how it can be applied to treat Zellweger spectrum disorder. I'll explain our efforts in Dr. Nancy Braverman's lab at McGill University to develop retinal gene therapy for PEX1-associated Zellweger spectrum disorder. Finally, I'll discuss the path to clinical trial for our AAV8 PEX1 gene therapy and give a brief update of where we are now. So let's start with what is gene therapy? Well, the FDA definition is that gene therapy is a technique that modifies a person's genes to treat or cure a disease. This can mean a lot of things. So gene therapies can work by several mechanisms. They can replace a disease-causing gene with a healthy copy of the gene. They can inactivate a disease-causing gene that is not functioning properly. They can introduce a new or modified gene into the body to help treat a disease. In addition, there are many different gene therapy products being studied and developed out there, both preclinically and in the clinic, and they can take one of two major forms. One of these forms is ex vivo, which means the modification is done outside of the body. These can be either, taking out a patient's cells, modifying them, reintroducing them to the patient. It can also mean introducing a therapeutic that's not DNA, for instance, into the body as well. And there can be in vivo gene therapy on the other side, which means that the genetic modification is made directly on the living person or the body, not outside of the body. So very exciting time, especially in the rare disease space. So let's talk about a little bit about what is our approach. Our current approach, it's called gene augmentation. So how this works is that a normal gene copy is inserted into a virus. Now, this isn't your typical natural virus that can cause harm or can cause diseases. It's a virus that's been engineered so that it's bare bones down to its minimum components just to infect the cell. So this virus cannot replicate and it cannot cause the typical symptoms that we experience when we contract viruses in our everyday life. We're really just using it as a tool to deliver that functional gene into the cell type of interest. So our therapeutic normal gene copy is inserted into the viral vector. This virus will enter the cell via membrane vesicle. So it's going to deliver its therapeutic gene, its normal gene, into the nucleus of the target cell. From there, the cell itself can produce a normal functioning protein using that therapeutic DNA as a template. And then this normal functioning protein can go on and perform its tasks as normal in the cell. So it should, in theory, recover the function that is deficient. Now, why this is an appropriate approach for monogenic diseases like ZSD, which means diseases that are caused by changes in one specific gene per individual, is that this approach addresses the genetic basis of disease instead of treating the downstream effects. So we're aiming to go right to the root and treat the cause of the ZSD in this case, which is the genetic defect. Another attractive aspect of this approach is that it gives the possibility of a single treatment, because that therapeutic gene that's delivered into the cell will express indefinitely, or we hope indefinitely. To go a little deeper on our current gene augmentation approach, and to explain really how this is applicable to the retina and how it's delivered, we start with our adeno-associated virus, or AAV serotype 8 in our case, which is a type of AAV that is well suited to delivering the cargo into the outer retina cells that are our target. So we start with this virus carrying the therapeutic gene, and it is delivered to the cells of interest through a subretinal injection. So this is done using a needle that is injected into the eye and will release the therapeutic agent into the subretinal space, which is the space between the retinal pigment epithelium and the photoreceptor cells. These are the cell types that we have found are primarily affected in ZSD, so we thought they would be the appropriate cell type to treat. I know this procedure looks a little bit scary, but it's actually a day procedure that's performed in the clinic. The patient is asleep when it happens, and they will go home the same day. So it's probably more invasive looking in the picture than what one would experience in real life. And as everyone here likely knows, there can be many organ systems affected in Zellweger spectrum disorder. So you might be wondering, why did we choose to focus on the retina? Well, one main reason when we first started this study is that the retina represents an ideal target for an approach like gene therapy, in that it's easily accessible. We can inject into the eye and we can actually have access to the cells that we're trying to treat because it's a small compartment that we can target with our approach. Equally, maybe more importantly, I would say, is that vision was prioritized for treatment by the patients and their families. We heard this many times at GFPD meetings and with consulting in consulting with the patients and their caregivers. And this is because although there may be multi-system manifestations in ZSD, retaining or recovering vision, as many of you can imagine firsthand, would improve communication, learning, mobility, autonomy, employment, so many things that affect quality of life for people with low vision. Another important consideration is that all people with PEX1 associated Zellweger Spectrum Disorder, which is our focus for this therapy, develop retinal degeneration. So they may not develop all of the other multi-system manifestations, but they do all lose their vision. Now, we started by addressing the PEX1 gene as it's the most common cause of ZSD. And so it seemed like that we had the tools ready to go. And because of its commonality, the most feasible cause of ZSD to target. But it's important to note that if this approach is successful and works for PEX1, that would provide precedent for using the same approach to target other PEX genes or other genes that cause peroxisomal disorders. The model that we used to start with to develop this therapy is our PEX1 G844D mouse model. So this mouse has the murine equivalent of the most common human ZSD allele. Many of you may have heard of PEX1 G843D. So in the mouse, it's G844D. This gene change is associated with milder disease. So there is some partial function left with this protein. And so the mice have a long enough lifespan and we're able to study them over a long period of time. This mouse model recapitulates many of the human disease features, such as small size, abnormal peroxisome metabolites, like the very long chain fatty acid elevation or the plasmalogen deficiency. They have liver disease that's been characterized by a few groups, including Dr. Lingzhao Chen in Dr. Braverman's group. And they also have retinal degeneration, so a progressive loss of visual function and retinal cells. And prior to testing therapies, the characterization of this retinal degeneration, both past and characterization that's still ongoing, paved the way for us to know how to design the optimal therapy for this disease. We started off by piloting what we call a proof of concept study. And in basically what this means, it's a study that provides evidence that a certain approach might work in humans. So it's kind of that first evidence that this might be a good idea. So we performed a proof of concept study and we used our AAV8 PEX1 vector to deliver the therapeutic gene. We actually delivered the human PEX1 gene into the PEX1 G844D mouse eye by subretinal injection. And we saw improvement in the mouse of peroxisome biochemical metabolites in the retina and also improved retinal function by electroretinogram or ERG. This work was done in collaboration with Dr. Jean Bennett's group at UPenn. She's been an amazing collaborator and consultant, really, for advising us on how to design the vectors and what type of experiments we should do. And also in collaboration with Dr. Joe Hacia at the University of Southern California. So these first studies were supported by the GFPD and the Win Mahaffey Foundation. So we're very grateful. The financial support to do a relatively small pilot project paved the way or opened the door for us to be able to attract a lot more resources. So these studies really paved the way for us to attract both venture capital and grant support that allowed the continuation of this project. We developed and redeveloped the maturation plan and completed the necessary experiments to support clinical translation. This meant preparing a rigorous preclinical efficacy data package using mice and also cell lines. We filed intellectual property with our respective universities, it's a multi-year process, so it's still ongoing in some jurisdictions. And we completed interinstitutional agreements among our three universities that were part of this invention. So really that first proof of concept study was only the beginning. The next phase of preclinical proof of concept studies included re-engineering a vector design that would actually be usable in humans. So we did that. We validated this new vector. We saw dose response in human and mouse cell lines. We optimized the dose that we used in the mice. And then finally, we performed a much larger long-term preclinical efficacy style in our PEX1 G8440 mouse model, with now armed with more outcome measures that we had refined so that they're less variable. So this time, we saw an even greater improvement in retinal function and retinal structure than we saw in our first studies. And I'll just give you a couple of small highlights of what those improvements were. So one important outcome that we looked at was whether functional vision improves in the mice. So it's all well and good if the peroxisome metabolites are corrected, but it's important to know if this actually translates to better vision in the mice. And visual acuity is in mice, the technique that we use to measure it is something called an optomotor reflex test. So the mice are placed in a chamber, and we purchased this platform at our lab at McGill, and they are challenged with black and white stripes that move around them, and the thickness of the stripes varies. And so the smaller the stripes are, the harder they are to see. The fatter they are, the easier they are to see. And the mouse will have a natural reflex to track something that's moving in front of its face. So we scored this visual acuity, or the mouse tracking, and until find the threshold at which they could perceive the stimulus. And this correlates to visual acuity. Another test that we could do with these stripes is contrast sensitivity. So in the first test, the bars were just black and white, but then you can have, instead of black, varying degrees of gray. So the lesser the contrast, that is, the lighter the black or gray bars are, the harder they are to perceive. And so when we look at the results here that are 6 months after the retinal gene therapy treatment, in the first bars, we have all the littermate control mice that do not have the PEX1 defect. So the first 4 bars are kind of the typical mice that do not have ZSD. On the right are the different mouse groups that do have the PEX1 G8440 gene change. So in black, they're untreated. In gray, they're treated only with the vehicle that's used to deliver the gene therapy. In green, they're treated with an AAV vector that has a different protein, a green fluorescent protein, that will express a green fluorescent protein, not the PEX1 protein of interest. But in blue are the mice that received the experimental therapy. So these in blue are the mice that received the gene therapy. So as you can see, the visual acuity in these PEX1 G844D mice drastically improves with the retinal gene therapy treatment. When we look at contrast sensitivity, again, we see a positive result in that the mice that were treated with the PEX1 retinal gene therapy have a lower contrast sensitivity or can perceive lower contrast, which is a good thing. So in this case, lower is better. That actually matches or is very close to mice that do not have the PEX1 gene defect at all. A second functional study that we did is an electroretinogram, so an ERG. This is a very similar technique as what's done clinically with human patients, but we were able to get this very special apparatus that is specifically tailored to murine subjects. And so the mice are put to sleep. So they're just put to sleep for about half an hour and put on this platform. And their eyes are stimulated with flashes of light of increasing intensity. And then these stimulators can measure the response of the retina to that light. And what we see, again, 6 months post-treatment is that, well, if you look at the bar here at the top, that's always the non-disease control mice. And in blue are the PEX1 G844D mice that were treated with gene therapy. So we can see a very stark improvement in their retinal response compared to the G844D mice that had the no treatment or other control treatments. And so the effects of both the visual acuity improvement and the ERG improvement were seen starting even at 2 months after gene therapy administration and continued to the experimental endpoint of 6 months. Another very interesting improvement that we saw, and these are all, these studies were done by Dr. Sami Omri in Dr. Nancy Braverman's lab, is that we see a preserved structure of the cone photoreceptors. So on the right here, we have mice that do not have any disease. And you can see that G844D mice lose this photoreceptor outer segment. But mice that are treated with the AAV8 PEX1 actually regrow this outer segment. So that was really amazing of the cones. We also see a preservation of the retinal pigment epithelium. Again, here is a normal case. Here is an example of a G844D mice. So you see the cells are becoming enlarged because we're losing cells, so they grow to fill the space. But in RPE that was treated with our gene therapy vector, we're able to preserve the structural integrity of those cells. So we were very pleased with these results. And so now it was time to actually prepare for an eventual clinical trial. So to get ready for that, there are several things that have to be put in place. First was looking forward to the question of how would one even design a clinical trial? And this topic, Frédéric Arnault touched on it in the last GFPD webinar. And so I won't go into detail there, but we reviewed ophthalmology data from patients that were enrolled in our lab's retrospective natural history study. And then that guided kind of the design of the prospective natural history study of vision in ZSD that is now being conducted both at our site at McGill and at Dr. Alex Levin's site in Rochester, New York. And so this study will allow for the design of an optimal clinical trial. Another item that will be helpful, especially when we look for more industry investment, is we put together prevalence and incidence estimates of intermediate to milder PEX1 associated ZSD in key countries, or at least in countries for which we have data that would allow for these types of estimates. So this allowed us to kind of have an idea of what the patient population would be and better address those questions if they come up from industry collaborators. Next, we prepared for and had our first preliminary meeting with a regulatory agency. So about a year ago now, we had our early pre-CTA with Health Canada. That is the equivalent of an FDA Interact meeting. So this involved submitting a briefing document that included all of our proof of concept data and also a proposed clinical trial enabling plan, which is basically a plan for how we're going to manufacture and support the safety of this intervention. They gave us a lot of feedback and guidance about topics like manufacturing and all the different safety studies one would have to do before considering a clinical trial. So things like how to design biodistribution studies, how to figure out the right dosing, what species to use for these safety studies, and also some clinical considerations to think about, like what population would one treat in a clinical trial. So armed with that meeting, our next step, or the results of that meeting, our next step was to set up clinical grade manufacturing. So what this means is that it's a very different process from reagents that we use for preclinical research. So we're now in the process where we must develop the methods and the strict, rigorous controls to produce a clinical grade product. So this process it will standardize the quality and the amount of AAV8 PEX1 produced. The processes for this are called good manufacturing processes and require very specialized facilities and expertise. And so we needed a partner or a service provider that could do this for us. It's not something that can be done in an academic center like a university most of the time. So we considered several different service providers, both in Canada and the United States. And after considering multiple factors, we decided to partner with a company called Vinta Bio. This is a Philadelphia-based contract development and manufacturing organization. Our decision was based both on the cost that they offered us, and also their collaborative nature, and that they were very helpful in helping us design the right manufacturing plan for us. So we expect to have our GMP-like vector batch this coming fall, if all goes well. So we're very excited about that. One fun fact is that one of the founders of Vinta, Junwei's son, was actually previously part of Dr. Bennett's research group at UPenn, and he was involved in actually providing our original research grade vector. So we're happy that it's like this project or this therapy is coming full circle in a way that the team is staying engaged and involved. And I will acknowledge the Canadian Institutes of Health Research for granting us, actually, we received a new grant specifically for accelerating rare disease gene therapies to clinical trial. So this manufacturing work, which is very costly, would not have been possible without this CIHR grant from the Canadian federal government. Our next step is really to preparing for clinical translation is to perform safety and toxicity studies. And these must be done in non-human primates. So again, these are very specialized types of experiments that cannot be done in an academic lab and also require service providers. There's two parts to these studies. One is a dose range finding study, which takes about a month. we'll primarily look at the effects in the eye of our gene therapy vector. And that helps narrow down what dose is, what we should use for the part two, which is the definitive toxicity studies, which take about 6 months or more to complete. And these definitive tox studies involve factors like biodistribution, meaning we look where in the body the AAV8 components and the therapeutic PEX1 gene are found. And we analyze different time points. We'll look at toxicity to different organs, just in case, to make sure there's no tissue damage or other toxic effects that occur after treatment. And these results will also help us predict an optimal therapeutic dose. So just like the manufacturing, these studies must be completed using good laboratory practices, which means the research is conducted following a set of guidelines that are more strict and different from typical academic research. So for this, we've met with several possible providers, and we hope to be able to raise the resources needed to pull the trigger and start on these studies too. So we've narrowed it down to an appropriate partner organization for these studies, but we do require more funds. My next slide is really just to give you a flavor of what's coming next for PEX1 retinal gene therapy development. As Katie mentioned at the beginning of the webinar, I will soon be moving on from McGill in Montreal and Dr. Nancy Braverman's group, which I've called home for a very long time, a great number of years now, and officially starting my independent research lab on August 1st. I will continue, above all, to champion the development and advancement of PEX1 retinal gene therapy. And my aim is to apply and hopefully be able to benefit from some of the unique supports that California has for regenerative medicine. And of course, I will continue my collaboration with Dr. Braverman. I'd like to acknowledge all my lab mates in Dr. Braverman's lab, especially Sami Omri and Armenia Di Pietro, that were instrumental in getting us to this point with our proof of concept work. Our collaborators, Dr. Hacia and Dr. Bennett at USC and UPenn, of course, all the funding agencies here that are critical for driving these types of projects forward. And I especially want to thank you, of course, for listening to me tonight. But I will reiterate that patient engagement is absolutely critical for rare disease therapy development. So thank you, patients and patient organizations and families, for your contributions. In terms of raising support, the fact that you're organized, that you have registries, that we can get letters of support from the GFPD, that you participate in the natural history studies makes our job a lot easier to convince granting agencies and industry partners this project is worthwhile and there's a true need for it. So thank you for your continued support. And with that, I'll turn it back over to Katie to moderate the question period.
Thank you so much. We're going to shift to Q&A, but I have to say just thank you again so much. We appreciate you sharing these remarkable updates with all of us and just for all of the work that you're doing and you continue to do. I know you mentioned, the vision loss and the impacts, but I just, wanted to reiterate that data collected even at the ELPFDD meeting in 2024, it's reinforced how profoundly vision loss impacts individuals with peroxisomal disorders. It's often ranked as the most impactful symptom. So your contributions are making a real difference, and for that, we are just so grateful. We're going to go ahead and move into some questions from the audience. You can continue to submit questions or comments you have by using the Q&A button option, which is below the webinar player window. And then also you can use the chat box to submit questions too. If anyone is more comfortable with submitting anonymously, that Q&A button does allow that option. One question that was submitted during the presentation, I think when you were mentioning the natural history study, was, how is this work connected to the prospective natural history study of vision in ZSD? Do you have to be a part of that to be considered for this trial in the future?
So to answer the last part of that question first, no, you do not need to be a part of that to be considered for this therapy in the future. Each study is completely voluntary and will not affect patient care or the possibility to participate at any later time. To go back to the first part of your question, the reason the prospective natural history study is very important for a therapy, especially in rare disease, to get to clinical trial is because the people designing the clinical trial, so we the scientists or the physicians in this case, need to know how to gauge whether or not the therapy is effective. So unless we have a picture of what changes in the eye, how fast the disease progresses, what are the different manifestations and what types of people, we don't know how to pick what are the best outcome measures to prove whether or not there's a therapeutic effect after the retinal gene therapy. So that's why it's very critical to collect this data because it gives us the best chance of success of designing the most accurate and robust clinical endpoints to show whether or not this therapy works if we get to clinical trial.
Thank you so much for taking the time to share all of that with us. And we also have more than one question a little bit on the variants and the different genes. So one is, would this therapy be specifically for those patients with PAX1 mutation or variant and not other PAX mutations?
So the short answer is yes. This therapy, because it's delivering a functional PEX1 gene to the retina, is specific to people with ZSD caused by PEX1 variants. So, to start with, this would be applicable to PEX1 ZSD. However, and so we chose this because it's the most common and so seemed like the kind of path of least resistance to actually getting support to get to the clinic. But if a technique works in one kind of ZSD, then it gives precedent or it gives enough evidence and makes it a little bit easier to then move to address other PEX genes or other PBD genes as well. So it kind of paves the way for that.
Thank you. And one of the ones that kind of came along with that would have been if that answer was yes, and it could be replicated, what would that time frame look like?
So I'd hope it'd be a lot faster because it would be, assuming the resources were there. I think the onus of providing the same amount of proof would be much less. We'd already have all the relationships in place. We probably might have an easier time proving safety. We have a manufacturing technique. And we have the clinical outcomes that would be the same for a lot of the different PEX genes. So I don't know what exactly the timeline would be, but I would assume and hope it would be a lot shorter than it has been for the PEX1 retinal gene therapy.
And many thank yous from the audience members who are asking those questions. We also have a question about, is there a certain level of retinal degeneration that one would have to qualify to be able to have gene therapy in the future?
So usually, yes. And that's kind of a loaded question in that typically when designing a clinical trial, the first people that will be treated are those with the most retinal degeneration, even though they may or may not have the same potential to respond to the therapy as someone earlier in the disease. So there would have to be a minimal amount of retinal degeneration to participate in clinical trial. And the therapy would be targeted to the right zones where there's still in the eye, where there still are cells left. So for clinical trial, yes, they would have to meet a threshold. of retinal degeneration. However, in the future, when this therapy is approved, if it is effective, then it could be applicable for any person with a PEX1 defect, kind of regardless of the stage of their retinal degeneration. So they could be treated earlier at kind of the earliest stages of their decline.
Thank you. And a few questions about the funding that you mentioned. What level of funding is necessary to advance this work? And if is the funding required now or for a future phase? Um, is it currently delayed? If you could speak to kind of what the urgency is of securing these funds.
Absolutely. So we have the funds to get through this phase of manufacturing that we've started. So the method development and a batch of virus that can be used for the safety studies. Now, we still need more funds to supplement doing the toxicity and safety studies in the non-human primate. For this, we're looking in the low millions US dollars for that step. And hopefully it will be feasible with different grant programs coming up. After that, we get to kind of regulatory approvals and clinical trials, which will require another set of funding. Again, there are grants available for these types of things. We hope by then maybe to have attracted another industry partner that would help. But it's like each incremental step requires funding. And so, yeah, we would right now need that ballpark $1 million US range to complete the safety and toxicity studies. And then that will buy us time to unlock the subsequent funds that we need.
Thank you. I think a lot of people had that interest and are very motivated by the work that you're doing. Um, do you think that this type of treatment could eventually help individuals with vision loss caused by etiologies other than peroxisomal disorders?
So this technology is or this approach or similar approaches are used to treat different retinal degenerations that are not related to peroxisome disease. So there is one retinal gene therapy that uses a different type of AAV to treat Leber's congenital amaurosis type 10. And so that is called Luxturna, and that therapy is widely approved in a lot of countries worldwide and is used clinically. So that paved the way for the rest of them. And there are several other retinal gene therapies in clinical trial at the moment for other inherited retinal degeneration, so other types of genetic diseases that result in vision loss. So we would not be the first using this approach, but we would be the first using it in kind of a multi-system metabolic disease like ZSD.
Great, thank you. And this may be a difficult one, I know. So we have to imagine the what ifs of if the funding continues to come through and each phase goes as hoped. What would be the timeline for the study to happen in humans?
Well, if we had the funding, right now, I think we could get through our safety and talk. If we had no financial barrier, I think we are lined up to do our safety studies within a year from now, and then maybe another half year to year to manufacture the next batch and get through regulatory approval for a clinical trial. So, if we had zero financial barriers, maybe 1.5 to 2 years, because our plan is really kind of lined up. But of course, it always is a bit slower or a lot slower than we'd like it to be because we have to always find the resources to do each next step.
I know that was going to be a hard one, so I appreciate you being very practical about that projected timeline, while also, providing hope that it's not too far off if the funding were to be there. There is another question that's pretty specific. We know that this is really specific to the retinas, but there are multiple diagnoses that people may have when they have vision loss with peroxisomal disorders. So We have someone who's asking if you could share a little bit about the tie-in with the very common diagnosis received of retinitis pigmentosa. How does this therapy help with that?
So retinitis pigmentosa is just part of the retinal degeneration or the vision loss in this disease. So that is a typical feature of the retinopathy that someone with ZSD will have. So that is really just a descriptive factor of losing certain cells or cell pockets in the retina, and is expected and is just a common feature of ZSD and other retinal degenerations. So it's not, something outside of what we would be looking to treat here.
Thank you. And I know you touched a little bit. I think this might be the last one, unless if anyone has any other questions or comments, please go ahead and submit them in that Q&A box or in the chat box. You talked a little bit about the natural history study, and someone wanted to know very specifically about, is the clinical data being collected by Frederick, who we recently just had that webinar on, is that being used to move this therapy forward?
Yes, it's directly related. So as that data comes in, and we're just in year 2 of a 5-year study there, but there is data already, and new patients are always welcome to join. Definitely still recruiting for that. That data is directly linked and important for designing a clinical trial for this gene therapy. So of course, it doesn't have to be data from people with PEX1 deficiencies. It can be any PEX gene that results in vision loss, but it feeds into the important factor of how we would test whether this therapy is effective and which patients we would predict would be appropriate to use in a clinical trial as well. As you know, people with ZSD can have a wide range of abilities and other complications in their life. And so we really need to get innovative with a clinical trial design that works for this unique disease. And that data directly informs how one would do that.
Thank you for diving a little bit deeper into how that, helps advance this work that you're doing. I'm going to go ahead and close out the Q&A if nobody else has any questions. Such great questions, though, and such a great presentation. Dr. Argyriou, thank you again for being here this evening, taking the time to share with our community, answer some of their questions, and just overall the vital work that you're doing to help those with peroxisomal disorders and vision loss. If you could move to that next slide, I just wanted to thank our audience as well for joining us. Remind everyone that this webinar recording, and the slides will be available on the GFPD YouTube channel soon. The vision webinar that we just had, in April is also available already on the YouTube channel. So if you missed that, I know it was referenced a few times tonight, you can go back and watch that before this one is released if you want to come back to this one. And then please make sure that you're staying up to date with us by visiting our website, following us on social media. If you're not receiving the GFPD Top 3 email newsletter, you can take a moment right now and you can scan the QR code that's on the screen, and that's going to take you right to a simple sign-up form, and you're going to start receiving important updates like the webinar we had tonight, the research that Dr. Argyriou is doing. So please make sure that you're receiving those top 3 emails. You can always reach out to me, Ryan, any of the members of the GFPD. You can contact us on the website. We will also ask that you please take a moment to complete the survey that will be sent to you after this webinar. Again, Dr. Argyriou and our audience, thank you, and I hope you all have a good rest of your evening. Thank you.
Thank you. Thank you, Katie, and everyone.